So today I'm going to talk about the role of oxygen in our biology and our genetics. Right, so let's start back 4.6 billion years ago. This is what primordial Earth looked like, not an actual picture. This is a video game representation. You can ask Paul Glover about what video game this is. And this is what Earth looks like today. So we see that there's a stark difference in the biodiversity. We have humans, we have fish, we have coral. And so the question is how did we make this jump and what biological adaptations had happened in order for us to get here? So at the formation of Earth, the atmosphere was very, very different. The gases were different, so helium and ammonia and methane, so you can imagine that organisms had to thrive in a very toxic environment, or what would be toxic to us. And then over the course of time here, there was some water vapor in the atmosphere. There's always been a lot of nitrogen present, and then carbon dioxide started to increase, and then it tailed off, and then finally, about 2.8 billion years ago, we started getting some oxygen in the atmosphere. So how did this happen, and how did this shape what we look like now? The oxygen has actually been around prior to 2.3 billion years ago. We had simple prokaryotic organisms converting CO2 into oxygen. Using the Calvin cycle, for example, CO2 was fixed to make sugars for the organism, and as a byproduct, oxygen, or O2, was released, and this was done by prokaryotic and eukaryotic organisms. However, the oxygen that was free was soaked up by minerals like iron, so even though there was oxygen being released, it was getting soaked up really fast by iron. So this is an old chunk of rock, and you can see how red it is, and that's the rusting of iron. And so it wasn't really until all of the iron and minerals had kind of been oxidized that there was free oxygen in the environment. And so that is when we start getting this uptick. So the red line represents kind of the upper limit of the estimation of oxygen in the atmosphere, and then the green is kind of the bottom, so it's kind of the standard deviation. So if we zoom more into our recent history, we see that there is a huge diversity in the amount of oxygen. So if this is today, we're around 20 or 21% oxygen, but 500 million years ago, we were half that, and then 300 million years ago, we were almost double that. So that has to have an impact on biology. We have to evolve to adapt to that. And so there's not just a diversity in oxygen levels throughout time, but on the planet, there's a large range of oxygen. So again, at sea level, we're around 20%, but as you go higher in elevation, when you're in Colorado, when you're in Mount Fuji in Japan, up to Mount Everest, you go from 20% oxygen to 7% oxygen. And there are peoples that live here, so how did they adapt to those environments? And so you might think that 7% oxygen must be really low, but your heart says otherwise. So even within our bodies, there's this huge dynamic range in O2 levels. So obviously your lungs are what take in oxygen, so they're going to be the greatest amount of oxygen will be concentrated there. And then your spleen also needs a lot of oxygen, but surprisingly, your brain, liver, kidney, and heart are only between 2 to 3% oxygen. And so the reason why oxygen is important to us is that it's a great electron acceptor. So even though nitrogen makes up around 78% of the gas in the atmosphere, and oxygen only makes up 20%, oxygen is the preferred electron acceptor because it's just intrinsically more reactive, whereas nitrogen is just intrinsically more stable. So all of our cells need oxygen to respire. We have mitochondria, which generate the cellular ATP that all of our cells in our bodies need to function. And so we have the inner membrane space where the electrons are moving through the different complexes, and pumping hydrogen into the intermembrane space, creating a concentration gradient to then power the ATPase to generate ATP. And ultimately, these oxygens need to come back down, and O2 is there to accept those oxygens. So O2 is forming water and accepting those electrons. So that's why oxygen is so important. And so when we think about why things look the way they look, a lot of it is in part because of oxygen and the necessity of all of our, the cells in our bodies require oxygen. So for insects, for example, they have adapted a very different system from us. So they don't have lungs, instead they have just one trachea that branches off into their tissues, and they actually don't breathe, they just use passive diffusion. And so instead of having just one mouth or nose the way that we have, they have multiple openings called spiracles. And they can actually open and close them, and they kind of push oxygen through to then oxygenate all of their tissues. So this is a passive diffusion event. So you can imagine that these bugs have to be small for a reason. They can't really grow very large because they require passive diffusion. On the other hand, dogs have evolved, like us, lungs instead of having a single or multiple openings. They just have one trachea with the two openings, the nose and the mouth. And what we see is that we have this great branching effect. And so the reason why the lungs are branching is to increase surface area. So if you were to envision in the same space just having two balloons, for example, the amount of oxygen that's touching the balloon on the inside, which would be your lungs, you wouldn't get very much oxygen transfer. Whereas in this case, when you have this great branching effect, essentially in the same volume, you're increasing the amount of surface area and you're optimizing the amount of oxygen that can be absorbed by the body. And so sea creatures are also doing this. So this is a salamander that's only found in a lake in Mexico, Oxalotl. And so this cute creature has these little things coming out here, and so it looks the way it looks because those are actually gills. And so again, we have this, if we zoom in closely, there is increase in surface area to promote the greatest amount of oxygen absorption. So again, you have this branching type of an effect in order to maximize the surface area. And so the plants on the planet also do the same thing, but for the opposite gas, for CO2. But they will have similar shapes to lungs, essentially, where you have branching, and then leaves themselves as a fern will also have some branching effects. So here's the primary part, and then you have a branch right here, and then off here you have those little guys. So this is all about gas exchange and optimization of that. And so here's what the human circulatory system looks like. Again, our lungs are similar to the dog's lungs, where we have the increased optimization of the surface area, and then we have a circulatory system, which then has branches off that to oxygenate the tissues and to bring nutrients to the tissues. And so this process is controlled very tightly by this oxygen sensing pathway. And so there are two key players. PHD2 is a propyl hydroxylase 2 enzyme, and this enzyme is what actually detects oxygen. So within the enzyme, in its catalytic core, oxygen needs to sit in there. So essentially, it's like a switch. When oxygen can sit in there during normoxic conditions, PHD2 can function to tag the Hif protein with OH groups. That's why it's called a propyl hydroxylase, which can then be identified by the ubiquitin ligase and then targeted for degradation. So under hypoxic conditions, PHD2 doesn't have oxygen within its core, and therefore it releases Hif from degradation, and instead it can act as a transcription factor. So the factors that Hif turns on have to do with pro-survival type factors. It releases EPO to tell the body to form more red blood cells. It turns on the glycolysis pathway. So if you're hypoxic and your mitochondria don't have oxygen, it makes sense to turn on glycolysis. And then it also releases angiogenic factors, VEGF, IL-8, ANG, to promote the growth of vesicles towards that area. So if you have an ischemic event, or if you get a wound or something and your tissue becomes poorly oxygenated, this pathway gets turned on and you get the formation of blood vessels in the right place. And so the balance of this pathway, you can imagine, is vitally important. You don't want to release too many or too little of these extracellular factors. And so one example of a genetically inherited condition that affects this pathway is cystic fibrosis. So cystic fibrosis, the protein for cystic fibrosis is CFTR, and it affects the stability of the HIF transcription factor. So normally under hypoxic conditions, or conditions when there's a lot of reactive oxygen species, HIF is stabilized and it can turn on its downstream targets. And so in CF patients, 22% of CF patients have anemia or subclinical anemia. So if you were to take a group of affected CF patients versus normal patients, they're always going to have lower levels of iron or hemoglobin. And so the severity of the anemia actually correlates with a poor outcome for CF patients. And so CF patients actually have a reduced capacity of erythropoiesis to generate more red blood cells. And so one model for why that is from this paper here, is that for some reason the mutant form of CFTR, or the knockdown of CFTR, generates a lot of glutathione, which kind of cleans up your reactive oxygen species. And as a consequence, HIF is no longer stabilized. So you get less of the HIF targets. And one of the HIF targets is EPO. So that makes sense why you don't have as good of erythropoiesis. You can also have the opposite effect, where you produce too many red blood cells, or polycythemia. And so if you are climbing a mountain, you're going higher and higher and higher, your body is going to change with the increase in altitude or the decrease in oxygen. So you'll start with having vision issues, you'll feel dizzy, your brain will start acting kind of funky, kind of slow, your memory will be poor, you'll hallucinate, you'll get loss of consciousness, and then you'll ultimately die. And so so the flip side of the HIF pathway is that it can actually be bad to have too much HIF turned on. So with polycythemia, you can end up getting hyperviscous blood, which can lead to pulmonary hypertension, cerebral hypoperfusion, and then eventually heart failure and death. And so the question is, if there are people on this planet that have adapted to living in these high altitude environments, what do they do, and how are they different from us? And so the Andean people in Peru, for example, they live in altitudes between 9,000 feet and 13,000 feet. And so on average, the amount of oxygen that's in their environment is around 12%. So these individuals, these natives here, are used to living in high altitudes, and they're totally fine at 12% oxygen. Whereas the gringos that go there on vacation have the opposite experience. And so the question is, like, do they look different? Can you guys tell anything about how they look? Is it just kind of their ethnic specific looks? I will say that that my experience there was that everything is labored when you're in that high altitude. It feels like you just did this crazy workout, and then you're expected to do this hike where each step is just so hard to take, and then your brain is slow, even though it's kind of euphoric at the same time. It's the weirdest experience. And so, and it's an experience that these individuals don't have because they've adapted to it. And so if you were to zoom in, what you would see is that these individuals actually have a large carotid body. And so they did this study, an autopsy study essentially, in 1976. And so individuals that are in the white bar are sea level individuals, and then individuals in the polka dot bars are the native Andeans. And so we're separating them into 3 groups based on age. Group A are the youngest group, Group B is 10 to 20-year-old, and then Group C is 21 to 40-year-olds. And so essentially, at birth, both groups of individuals have the same size and weight. So the length, width, height of the carotid body and the weight is essentially the same. But as both populations age, the population that's at the sea level, their carotid body stays the same, whereas the size and the weight of the Andean people, their carotid bodies increase with age. And so the model is that so the carotid body communicates with your central nervous system. So this is the brain part of the high altitude. So a small carotid body communicates with neurons, and you get this slow firing event. That's what makes it feel slow. Whereas a large carotid body has the capacity to increase the amount of neuronal firing. So this is kind of normalizing these individuals. And so the crazy thing about this is that this isn't genetic. This is a physical adaptation. So when they put rats in a hypoxia chamber, and they just left them in the chamber, and then they would pull out a rat and do a necropsy on them. Initially, the rats had very small carotid bodies, but then over time, over 100 days of hypoxic exposure, their carotid bodies increased. So the adaptation in this case is not genetic, but it's just triggered by the hypoxia. And across the planet, at a similar altitude, there's a different group of people, the Tibetan people. They also live in high altitude in the same range, 14,000 feet. And this is what they look like, and these are the Himalayas. What's interesting about them is that they don't have larger carotid bodies. They do have genetic alteration. So in this study, they took a cohort of 100 Tibetan individuals, and then they did whole exome sequencing, and then they also compared the results to the Han Chinese people and Danish Europeans. So if you imagine how people came out of Africa, they moved across Europe and then into Asia, this is kind of the trajectory that we're following. So they tried to identify variants that were specific to the Tibetan people that were either not present in the Danish or the Han Chinese people, or at a very low frequency. And so their top candidate genes were here, but then these genes were actually nearby other genes that they call their candidate genes. And so EPAS1, which is the Hif protein, was their first hit, and then there were a couple ORFs that were near another gene called EGLN1, and then several of the genes were near HFE, which is the hemochromatosis gene. And so using this data, they were able to draw kind of an evolutionary type of a line. So the distance between these lines kind of show the amount of genetic similarities. So the Han Chinese are genetically more similar to Danish people than the Tibetan people, which are kind of their separate population. And so what do EGLN1 and EPAS do? They are controlling the Hif pathway. So EGLN1 encodes PHD2, which senses the oxygen. So the Tibetan people actually sense oxygen different than we do. So the variant that is in here is where the oxygen binds. And so they actually have to go into a much more hypoxic environment in order for their PHD2 to respond to the change. And then once the PHD2 has responded to the change and released Hif is actually targeting different genes. So the second change in EPAS1 is affecting the transcription of a different group of genes. So not only are they sensing oxygen differently, they're responding to oxygen differently. And so what's really cool is that we can track these down through evolution. So the EGLN1 change was introduced very recently, about 8,000 years ago. And so from an evolutionary standpoint, when we think about genetic changes, we normally think in the millions of years, how you have a slow drift. But this is actually the evidence of the most recent evolutionary trait, adaptive trait in natural selection that we've noted. And so this EPAS1 variant has an interesting story. So once they discovered that this EPAS1 variant was specific to the Tibetan people and not present in the Han Chinese or the Danish people, they compared that position to all species. including like ancient Neanderthals. And so what they found was that there was one species called the Denisovian people that had the same variant. And so there's a cave called the Denisovian cave where they found like a fragment of a finger of an ancient people, kind of like the way we find Neanderthal fragments. And they did whole exome sequencing on that population. They think it's a distinct species. And so what this kind of shows is that kind of like how the Homo sapiens made it with the Neanderthals, they must have also made it with the Denisovian people in order to get this variant in their population. And so we have no idea what the Denisovian people look like. We only have that one finger, but we definitely know what Tibetan people look like. And so this is kind of what the genetic data shows. And so what's really interesting is that in the folklore of the origin story of the Tibetan people, they believe that their people were born because there was a massive flood in Tibet that wiped everyone out. And one of their ancestors, Pha Treigen Changchup Sempa, got reincarnated into a male monkey. That was this mythical monkey that had very even temperament, very monk-like, very Buddhist-like, who was lured by a female ogre sitting on a rock to mate with him. And then they had 6 children, some of which had similar temperaments to him, and some of which had similar temperaments to the ogre type of temperament. And so basically their origin story is kind of consistent with what the genetic data is showing. All right, so that's kind of how we adapted to oxygen in terms of adapting to high altitude. On a cellular level, there's a flip side to oxygen. So oxygen is damaging our DNA. So this is kind of what we have to deal with in order to enjoy being here on this planet. And so the formation of free radicals will damage our DNA. And so in one day, we get about 11,500 pieces of damage to our DNA. And so if we breathe around 23,000 times per day, one cell is getting 1 piece of DNA damage for every 2 breaths. So if we all breathe 2 times, you just had 1 damage to your DNA. And so one quote is that as long as we breathe, we use oxygen, and that rips up our DNA. It's like rusting of iron. And so we should celebrate that in 2015, so very recently, the Nobel Prize in Chemistry was awarded to these 3 individuals that described how we respond to that DNA damage. So DNA damage can be caused by oxygen, free radicals, by replication errors, by UV light, by X-rays, and there are several pathways that are used to repair those. So for oxygen specifically, we have evolved pathways for base excision repair, where you remove the faulty base, and then you can fill it back in with the correct nucleotide using the strand opposite. And then the other pathways are the mismatch repair, nucleotide excision repair, and homologous recombination. But then what we know in terms of genetic changes is that alterations in these genes or in these pathways, or if you inherit a bad copy of any of these repair pathways, this leads to inherited forms of cancer. So we have the BRCA pathway, which is involved in hereditary breast and ovarian cancer. We have the MMR pathway, which would be Lynch syndrome. ATR and ATM, which are the molecules that actually sense DNA breaks, that will give you ataxia telangiectasia, Lee-Fermini for P53. So alterations in this pathway have evolved to adapt to DNA damage. However, if you're deficient in this is when you get the hereditary cancers. And so, the first step in cancer formation is always some sort of genomic instability. And we know that oxygen plays a role in that by creating DNA damage through reactive oxygen species. However, oxygen also has a secondary role in tumor formation, and that has to do with the vascularization of solid tumors. So 90% of tumors are solid tumors, the other 10% are the leukemias. And so essentially 90%, or all tumors almost, are having to cope with vascularization issues. So if you were to have a tumor that just growed and growed and growed, ultimately it would outgrow its blood supply and nutrition supply. And so that would be a good thing because they would just become necrotic and then the body would clean them up. However, tumors have co-opted the HIF pathway. So this pathway, which is normal in development and wound healing, says release HIF, I'm hypoxic, releasing angiogenic cytokines as VEGF, IL-8, IL-2, A and G, and to grow more blood vessels towards me. And so tumors themselves are taking advantage of this pathway. So the cells that are close to the vasculature have a lot of oxygen, so they have very low levels of HIF, whereas these cells here that you would just hope that they would become necrotic and they would die, those are hypoxic and have high levels of HIF, and then they will release the pro-angiogenic factors, and then the blood vessels will grow into them, giving them oxygen and giving them nutrients, and that's why things like Avastin, the drugs that are going against the HIF targets, have been very useful in cancer treatment. And so now that we know that oh, I forgot one thing. And so I should note here that this VHL protein is the von Hippel-Lindau inherited tumor syndrome. So in this case, if you have a tumor where you have the loss of the VHL gene or the VHL protein, every single one of these cells thinks it's hypoxic and is releasing all these pro-survival, pro-angiogenic factors. So if we look at the tumors themselves, and if we perform some immunohistochemistry, we learn that high levels of HIF is a poor prognosis. So this is a tumor microarray. It's similar to a DNA microarray, but instead of having DNA on a slide, you have pieces of tumors on the slide, and then you can do immunohistochemistry on several tumors, like 80-some tumors at one time. And so this is an example where you have a tumor that stains very weakly for the HIF1 protein. So the HIF protein is recognized by an antibody, and then essentially you turn the antibody into a signal. So the amount of brown staining corresponds to the amount of HIF1 that's in that tumor. So if you have very little staining, the patients with this amount of HIF1 in their tumors, they can survive more than 10 years post-surgery. However, patients whose tumors have high levels of HIF1 staining have a very poor prognosis and live less than a year. And so this is a pancreatic tumor microarray. And so if you were to separate out patients whose tumors have high HIF expression versus low HIF expression and do a survival curve, so on the x-axis is the number of years that the cohort survives after surgery, and the group in black are the patients whose tumors have very low staining of HIF, and then the patients that are in red are the ones who have very high staining of HIF. You can see that, like, after 6 years, for example, you have almost 30% survival of the low HIF staining group, whereas you have a very rapid decline in the patients that have the high HIF staining. And this isn't specific to just pancreatic cancers, because all solid tumors are going to be taking advantage of this pathway. We see that using the Kaplan-Meier curves, you can separate out the patients using HIF, and you can predict their prognosis for soft tissue carcinoma, breast cancer, and esophageal cancer. Essentially, every solid cancer will do this. And so when we think about the oxygen response pathway, we kind of only think about multicellular systems. So if we are in our bodies, our whole entire body wants to survive, and all the cells want to survive, it makes sense that you have to communicate with one another in order to achieve the proper homeostasis and the oxygen flowing and the nutrients. However, what's really amazing is that unicellular organisms also can do this. So in the lab, when we think about bacteria and yeast, we normally think of them looking like this, where they're just an individual colony of cells, and that's kind of all they do, right? However, recently it was described that the unicellular organisms can communicate with each other to form these crazy 3D structures. And so this is kind of what we think of as a normal colony. Like after 2 or 3 days, you just throw the plate away. But if you were to let the plate sit another day, you'd start getting these growths that are growing upwards, kind of this biofilm type of a formation. However, if you were to give them in an environment where there's extra oxygen, you lose that formation of that 3D structure. And so in this paper, they did this crazy thing where they built this tiny little probe that can measure the amount of oxygen. And then if you place the probe on top of this colony, it's very lowly oxygenated. But then if you put the same probe on top of these colonies into those little valleys, it's oxygenated very, very well. And so it seems like this is an adaptation to oxygen and to increase the amount of surface area, the same ways that our lungs are trying to increase the amount of surface area to get the maximal amount of oxygen exchange. And so these authors also did the flip side of the experiment, where these cells were grown in slightly reduced oxygen concentrations, and you get the formation of those 3D valleys and mountains much earlier. And so at day 4, for example, you get an even more striking pattern of the colony formation. And so I'm going to leave you with just some pretty pictures. So this work was done by the Dietrich Lab at Columbia University, and they just posted these pictures on their website just to show the amount of diversity that can happen in the formation of the mountains and the valleys, all of which are to increase the amount of surface area. And so I was kind of inspired by this, and like after I made this talk, I kind of realized that like just in our environment, we can see a lot of these trends that I was talking about, and we can actually see it in the GDC parking lot even. And so you can think about cells like cars and how people park. And so when the parking lot isn't very full, you don't have to be organized very well, right? You can have one car really close to another car. So if you look at this line right here, that's the actual line for the parking stall. And like the tire is completely over with a gap even. But if you have like kids in the car, you can roll down the windows, and you could roll down this car on the left, you can roll down the left side of the windows, and then this car on the right, you could open the right side of the windows, and they would be okay. However, when the parking lot is more full and when you have to communicate with one another, you have to get more organized, and then you have to create a better structure, and you have to organize and park better. And if I'm not driving the point home, all right, that's it.